For decades, the story of complex life seemed clear. Oxygen was the key. It fueled the rise of animals, plants, and fungi. These organisms, known as eukaryotes, possess complex cells with a nucleus and other specialized compartments. This complexity, we thought, demanded the energetic boost of oxygen. But science is a journey of constant discovery. A recent study from China now whispers a different tale. It suggests that the earliest eukaryotes did not depend on oxygen but might have thrived on hydrogen. This forces us to reconsider the fundamental requirements for complex life. To understand this new theory, we must travel back in time. Billions of years ago, Earth was a very different place with an atmosphere thick with gases like methane, ammonia, carbon dioxide, and hydrogen. Life existed, but it was simple, dominated by bacteria and archaea. Then came the Great Oxidation Event, roughly 2.4 billion years ago, driven by cyanobacteria. Slowly, oxygen began to accumulate, triggering a mass extinction but also paving the way for new evolutionary pathways. The traditional view holds that eukaryotes could only have emerged after oxygen became plentiful, but evidence suggests that pockets of anoxic environments persisted long after the Great Oxidation Event. It is in these hidden corners that the hydrogen hypothesis finds fertile ground. This new study forces us to look beyond the oxygen-centric view. The hydrogen hypothesis offers a compelling alternative to the oxygen-centric view of eukaryotic origins. This hypothesis shifts the focus from oxygen, which is often considered essential for complex life, to hydrogen, a simpler and more ancient element. It was first proposed in the late 1990s by William Martin and Miklos Müller. Their groundbreaking work challenged the traditional views and opened new avenues for research into the origins of complex life. The core idea is simple yet profound. The first eukaryotic cell arose from a symbiotic relationship centered around hydrogen. This relationship was not just a random occurrence, but a pivotal event in the history of life on Earth. An archaeal host cell, one that consumed hydrogen, engulfed a bacterium that produced hydrogen as a waste product. This event marked the beginning of a new era in cellular evolution. This created a perfect metabolic partnership. The archaeal host and the bacterium became interdependent, each benefiting from the other's presence. The bacterium provided a steady supply of hydrogen to its host, which in return provided a safe environment. This mutualistic relationship was crucial for the survival and evolution of both organisms. This intimate association was the crucial first step leading to the formation of the eukaryotic cell. Over time, this relationship became more complex and integrated, paving the way for the development of more advanced cellular structures. The engulfed bacterium eventually evolved into mitochondria. These organelles became the powerhouses of the cell, providing energy for various cellular processes. Initially, mitochondria were hydrogen-producing factories for their archaeal host. This function was essential in an anoxic or oxygen-free environment. Only later, as oxygen levels rose, did mitochondria adapt to use oxygen. This adaptation was a significant evolutionary step, allowing cells to harness more energy and become more complex. This reinterpretation of mitochondrial origins is central to the new Chinese study. The research provides fresh insights into how these organelles evolved and adapted over time. It provides a framework for understanding how complex cells could have emerged in an anoxic world. This new perspective not only enriches our knowledge of cellular evolution, but also highlights the incredible adaptability of life on Earth. The search for our earliest ancestors has led scientists to a fascinating group of microbes called Asgard Archaea. These tiny organisms have opened a new window into understanding the origins of complex life on Earth. These organisms were first discovered in deep-sea sediments, where they thrive in extreme environments. The discovery was groundbreaking, revealing a hidden world beneath the ocean floor. Asgard Archaea are prokaryotes like bacteria, but their genomes possess a surprising number of genes previously thought to be unique to eukaryotes. This finding has challenged our understanding of the tree of life. These eukaryotic signature proteins are involved in complex cellular processes such as cell division and intracellular transport. Their presence in Archaea suggests a more intricate evolutionary history than previously imagined. Finding these genes in Archaea suggested a close evolutionary relationship between Asgard Archaea and eukaryotes. This connection has profound implications for our understanding of how complex cells evolve. Many scientists now believe that Asgard Archaea are our closest living prokaryotic relatives. This relationship provides a crucial link in the evolutionary chain, bridging the gap between simple and complex life forms. The Chinese study focused heavily on these Asgard Archaea, analyzing their genomes to reconstruct aspects of their ancient lifestyle. By understanding their genetic makeup, researchers hope to uncover the secrets of early life on Earth. The presence of eukaryotic-like genes in Asgard Archaea strengthens the idea that the host cell in the original symbiosis was indeed an archaeon. This symbiotic relationship may have been a pivotal moment in the evolution of complex life. These microbes are key to testing the hydrogen hypothesis, which suggests that hydrogen production played a crucial role in the early evolution of life. By studying Asgard Archaea, scientists hope to unlock new insights into this ancient process. The recent study by Chinese scientists employed sophisticated genomic analysis. They delved deep into the genetic makeup of Asgard Archaea to find clues about the metabolism of the ancient archaeal host. The researchers examined entire genomes, comparing them to the genomes of modern eukaryotes. This comparative approach allows scientists to identify shared genes and trace their evolutionary history. They specifically searched for genes related to hydrogen metabolism. The hydrogen hypothesis predicts that the archaeal host was a hydrogen consumer. Therefore, its genome should contain genes for enzymes that process hydrogen. The study meticulously scanned the Asgard archaeal genomes for these telltale signs. The thoroughness of this genomic investigation is what makes the study so compelling. The findings of the Chinese study are striking, revealing new insights into the ancient world of microorganisms. The genomic analysis of Asgard Archaea revealed compelling evidence that these microbes possess the genetic machinery to utilize hydrogen, a crucial element in their survival. Specifically, the researchers identified genes for hydrogenases, enzymes that can process molecular hydrogen, which is a significant finding in understanding microbial life. This discovery supports the idea that the archaeal host of the primordial eukaryotic cell was indeed a hydrogen consumer, playing a vital role in early cellular evolution. Moreover, the study provided insights into the nature of the bacterial symbiont, suggesting it was capable of producing hydrogen, which would have been essential for the archaeal host. This mutual dependency would have driven the two organisms into an increasingly intimate and permanent association, forming a symbiotic relationship that was crucial for their survival. The research also sheds light on the timing of these events, suggesting that hydrogen metabolism was an ancient feature of the Asgard archaeal lineage, dating back billions of years. This strengthens the argument that the origin of eukaryotes occurred in an anoxic setting, fueled by hydrogen, which was abundant in the early Earth's environment. The later acquisition of aerobic respiration was an opportunistic upgrade, allowing eukaryotes to thrive in a newly oxygenated world, marking a significant evolutionary milestone. The implications of this research for understanding early life are profound. It suggests that the evolution of cellular complexity did not require oxygen. This challenges a long-standing paradigm in biology. For many years, scientists believed that the high energy demands of complex cells could only be met by aerobic respiration. This new study, however, shows that a hydrogen-based metabolism could have provided sufficient energy. This finding opens up new possibilities for where and when eukaryotes might have first appeared. If oxygen was not a prerequisite, then complex life could have originated much earlier than previously thought. It could have emerged in diverse anoxic environments such as deep-sea hydrothermal vents or anoxic sediments. The traditional focus on oxygen-rich surface waters as the cradle of eukaryotic life may need to be reconsidered. The discovery that early complex life on Earth might have thrived on hydrogen has exciting implications for astrobiology. It significantly broadens the range of environments where we might expect to find life, even complex life, beyond our planet. For a long time, the search for extraterrestrial life has been guided by the "follow the water" mantra. Oxygen has also been considered a key biosignature. However, this new research challenges that assumption. If eukaryotes could emerge and flourish using hydrogen in anoxic conditions, then similar processes could occur on other worlds. Many celestial bodies in our solar system and beyond are thought to have anoxic environments. Some may possess subsurface oceans or hydrothermal vent systems where hydrogen is abundant. This new perspective means that an oxygen-rich atmosphere might not be a prerequisite for complex life. The Chinese study on hydrogen-breathing ancestors is more than just a single discovery. It is a testament to the dynamic nature of science. Our understanding of life's history is constantly evolving. New tools like advanced genomic sequencing allow us to peer deeper into the past than ever before. The idea that our distant forebears were powered by hydrogen, not oxygen, is one such surprise. This research has far-reaching implications for evolutionary biology. It forces a re-evaluation of the key steps that led to the emergence of complex life. The timing, the environmental context, and the metabolic drivers of eukaryotic origins are all being re-examined. The story of evolution is not a fixed narrative. It is a living field of